Welcome to my channel. Today I will discuss about enzyme. What is enzyme? Enzyme is a biocatalyst. That means they have the capability to catalyze the biochemical reaction. Here is enzyme and it must have a substrate, and this enzyme substrate they make complex and then they produce the product. And enzyme, they have three criteria. First, they accelerated the rate of reaction. That means they accelerated reaction rate, and after that it itself remain unchanged, and it required very small amount. So to be an enzyme, it must have the capability to accelerate the reaction and also itself remain unchanged, and also it need very small amount. Commonly, all enzymes are protein, but there is some exception. A small group of RNA molecules which have the catalytical ability, this known as ribozyme. What type of catalytical activity? They accelerate the reaction rate and itself remain unchanged and need very small amount. And as the nature of like protein, enzyme also colloidal water soluble heat level. That means if we increase the heat, it denature occur and it became non-functional as like protein. And all enzymes have active sites. That means enzyme must have active site. And enzyme has unique properties like specificity towards substrate, and that has enormous catalytic power. Let's dig into details. They don't initiate the reaction. They never initiate reaction, but they accelerate the reaction. They only accelerate the reaction, and the acceleration rate typically 10 to the power 6 to 10 to the power 11 times, and sometimes more, to reach the equilibrium. And they don't shorten, but they quick. They make it quicker. And they don't alter the equilibrium constant of a reaction, bear in mind, and they're highly substrate specific. That means what is substrate? Substance on which enzyme work to produce product, that not as substrate. And enzymes are highly specific towards substrate, and they are effective in extremely small quantity, very few. It need very few to complete it mechanism process. And activity can be regulated. The enzyme's activity can be regulated by activation and also through the inhibition as per cellular need. On the basis of cellular need, we can activate enzymes activity or we can inhibit enzymes activity. And as I told, is a sensitivity to heat, that is high temperature make denaturation, and also it's specific to pH. Like each catalyst is most active at a particular pH. For example, in case of pepsin, the pH, appropriate pH is 2, and in case of trypsin 8.5. Let's talk about enzyme activity energy. Look at this graph. Here these are the substrate or reactant and these are the product. So the substrate must reach in this position. How they reach? Without enzyme, they first activate their reaction and come in a transition state, and then go down to free energy change, and then finally come toward the products. But with the help of enzymes, that is with the help of enzymes, this activation energy lower in this type. No need to go here. So it actually reduce the activation energy, and then it reach toward the products. So enzyme activation energy, what is it? Lower the energy barrier to speed up chemical reaction. So enzyme helps to lower the activation energy from this portion to here. And without an enzyme, reaction require high energy activation. As I showed, high energy activation without enzyme, without the help of enzyme, activation energy is higher. Why? Because it needs sufficient force and proper orientation to breaking down the bonds between the chemicals. And also With the help of an enzyme, they can stabilize this transitional stage, and they can show alternative reaction pathway and also optimum alignment. They bring the substance in optimal alignment so that the enzyme activation energy lowered, with the help of enzyme activation energy lower, and which make it faster. So how actually enzyme helps to catalysis, and how they reduce the activation energy? There are three main catalytic mechanisms. One is acid base catalysis, another one is covalent catalysis, another one is metal ion catalysis. Let's talk about acid base catalysis. Acid base catalysis, here the mechanism is so simple. Enzyme donates or accepts a proton, that is protonation and deprotonation occur here to stabilize a reaction intermediate. They try to stabilize a reaction intermediate by donate or by accept proton. For example, ribonuclease A, or we can call shortly it RNase A. How RNase A occur? Helps to acid base catalysis. RNase A cleaves RNA by using histidine residue. So RNase A cleaves RNA by using histidine residue, and this histidine residue can act as proton donor and acceptor. Let's talk about covalent analysis. Covalent analysis, here's the mechanism also simple. Enzymes forms a temporary covalent bond with the substrate. That means enzyme form a temporary covalent substance with the substrate, and the example is here chymotrypsin. For example, in chymotrypsin is a serine protease. Why it's serine protease? Because in its active site serine residue present there, and they are actively play a vital role to attack the peptide bond, forming a covalent acyl enzyme intermediate, which is later hydrolyzed to release the product. That's how covalent catalysis work. What about metal ion catalysis? Metal ion catalysis, or we can call it another way, cofactor assisted. Here the mechanism is metal ion stabilize negative charges which assist in electron transfer or help orient substrate. For example, carbonic anhydrous. Carbonic anhydrous, here zinc ion bind in the active site, and it helps to polarize water to generate hydroxide, which then attacks carbon dioxide and to form bicarbonate. And what about transition state of an substrate? It's unstable, but here enzyme helps to stabilize the transitional state or intermediate state of enzymes of the product and substrate complex. Here Linux Pauling explain it details. I will discuss it later in another video maybe. Let's go into the structure of an enzymes. Structure enzyme, as I told, that it must have active site. This one is active site, this is active site, and this active site is the region that bind the substrate. This is the substrate, and this one enzyme, and enzyme has active site, and this active site here the substrate will come and they bind and make a complex. An active site is occupy only less than 5% of total surface of an area. You have seen here the total enzyme and the active site is very lower portion, only the 5% or less than 5% even. And active site has a specific shape, as you have seen for the example in case of this one amylose, and this is amylase active on catalyze on amylose, that's why it's called amylase. And here the active site shape is very similar with the substrate, that is the amylose, so that amylose can easily fit here, fit with the active site. That means active site has a specific shape due to the tertiary structure of protein, and in the residues near the active sites are usually histidine, aspartate glutamate, serine, cysteine, lysine, threonine or arginine. And a change of the active site affect the protein site, and it also affect the function of that enzyme. So active site is very important. Let's talk about Zymogen. What is Zymogen? Zymogen is an inactive precursor form of an enzyme. That means an enzyme is in an inactive form that can be activated to be a full functional enzyme by some modification. How it's possible? By proteolytic cleavage, that is cutting off a short peptide segment. For example, trypsinogen. Trypsinogen is a zymogen, and by the cleavage of little bit of peptides, it form trypsin. Trypsin is an active form of an enzyme, and by changing the pH, like for example pepsin is inactive in high pH, but when the pH is lower, like activated in stomach where pH is lower, 2.2 or something, then it became active. And also by binding to another molecules, that is the cofactor or protein partner, that can also make the zymogen in activated enzyme. For example, DNA polymerase, it requires magnesium ion as a cofactor to be functional. So zymogen is an inactive precursor form of an enzyme, and it can be activated by proteolytic cleavage or pH change or binding to another molecules. In mammals, for example, pepsinogen is the zymogen form, inactive form. An active form is pepsin. Trypsinogen, inactive zymogen, which can convert to trypsin, activated form of an enzyme. Chymotrypsin, chymotrypsinogen, inactive form, zymogen, which can be converted to chymotrypsin. And also fibrinogen, fibrin, prothrombin in thrombin. They are blood clotting, related to blood clotting. And in case of plants, protease zymogens. Maximum in plants proteases are zymogens. For example, cysteine proteases and serine proteases. As I told, what is cysteine proteases? Because in this enzyme the inactive site residue is cysteine, and also in case of serine protease, these are serine, and they are synthesized as zymogen. That means they are in inactive form. Like for example, one cysteine protease is propapain. Propapain is a zymogen, and in little bit cleavage of this propapain make it active form of papain. Papain found in papaya. And also same thing in case of probromelin. Probromelin is an inactive form of enzyme, that means the zymogen, and it can be activated to bromelin, which is active form of enzyme, and it's found in pineapple. Let's see another example, activation of chymotrypsin. For example, chymotrypsin, to activate it needs trypsin. It's interesting. To activate chymotrypsin, it need trypsin. Chymotrypsinogen, it's zymogen, inactive form, and then in cleave little bit, and it make pi chymotrypsin. This is active form. Here cleavage occur, and then the removal of little bit part of serine and arginine, serine 14 and arginine 15, with the help of chymotrypsin. Then here the missing of 13 and 14 and 15, that is serine and arginine, and this one is delta chymotrypsin. This is also active form of chymotrypsin. And then again in little bit cleavage of 147 threonine and 148 asparagine. Here this cleavage also make it another active form, that is the alpha chymotrypsin. This alpha chymotrypsin, delta chymotrypsin, and also pi chymotrypsin, these three are active form of chymotrypsin, but this one is chymotrypsinogen, inactive form. And how these peptides are joined with each other? They are joined by each other with disulfide bonds. Let's talk about temperature coefficient of enzyme. Temperature coefficient of enzyme, this is the change of relative reaction rate, change of reaction rate of enzymes at two temperature difference, that is the temperature difference by 10 degrees Celsius. That means in every 10 degree Celsius temperature, the reaction rate of an enzyme known as temperature coefficient. Look at this graph. Here the ratio of this R2 and R1, R2 is reaction rate 2 and R1 is reaction 1, and here the difference of the temperature, and here we can find the temperature coefficient or Q10 for enzyme. How it calculate? Q10, the formula is Q10 equal R2 by R1. R2 stand for reaction rate at temperature T2, R1 is reaction rate at temperature T1, and 10 divided by T2 minus T1. T2 is temperature at T2 and the primary temperature T1. For example, suppose an enzyme has a reaction rate of 50 micromole per minute at 20 degrees Celsius. So here R1 is 50 micromolar, and T1 is also 20 degrees Celsius, and another is R2 enzyme rate 100 micromolar per minute. This one is R2 and at 30 degrees Celsius, that means T2. So how we can calculate this? So Q10 will be R2 is 100 divided by R1, that is the 50 whole to the power 10 degrees Celsius, and for temperature T2 30 minus temperature T1 20 equal 2. So the reaction rate doubles with the 10 degrees Celsius rise. Now let's talk about enzyme portions. So in enzyme, the protein, it consists protein part. This protein part known as apoenzyme, and also it has some non-protein parts. These are non-protein parts. They are called cofactors, and these non-protein cofactors may be organic or inorganic. If it's organic, then we call it coenzyme, and if it's organic or inorganic, then it's total cofactor, as I told. And what is holoenzyme? Holoenzyme is total protein part and non-protein part. These are non-protein part of an enzyme. Let's talk about cofactor. As I told, cofactor is chemically compound, and they may be organic or inorganic. Chemical compound means inorganic, and they might be organic sometimes, which we call coenzyme. And these are the some example of inorganic ions. For example, cupric ion in case of cytochrome oxidase. In case of cytochrome oxidase, they act as cofactor. Magnesium in case of hexokinase, glucose 6-phosphate, etc. And these are the inactive portion of, that is the apoenzyme protein part. Is this one is inactive. So cofactor add here to make space to create for the to make binding site for the substrate to fit. That's how it cofactor make this inactive protein active, so that they can easily bind with the substrate. Let's talk about prosthetic group and coenzyme. Again, prosthetic group may be organic or inorganic, and they are tightly bound with covalently, sometimes covalently bound, and sometimes even permanent binding. What about coenzyme? Coenzyme is organic. Bear in mind, only coenzyme is the organic cofactor, and they are loosely attached. So here there is a coenzyme, what is organic, and they are loosely attached with the protein part of an enzyme. And in case of prosthetic group, they are tightly attached, and they may be organic or inorganic. For example, FAD, and in case of coenzyme like NAD, for example. Let's talk about the mechanism of enzyme action, how enzyme work. There are two models so far introduced. First one is lock and key hypothesis, introduced by Emil Fischer in 1894. And in case of lock and key hypothesis, the active site will be fixed, like lock and key. It's fixed, it's rigid, and substrate must have the same structure with the active site. Then it's like lock and key. Key must be the fixed, and lock hole also must be at the same pattern like the key. So in this case, active site is rigid. This is the take home message. And what about induced fit model? Another model, induced fit model, introduced by Coastland in 1958 recently. Here the active site is flexible. That means the enzymes active site here, there the amino acid residue can be alter each other, or they can make the comfort zone for the substrate to enter. So they are flexible, they are not rigid. The shape of enzyme of active site and substrate they adjust with each other to maximize the fit, and which improve catalysis. Let's talk about the nomenclature of enzymes, how we can nomenclature the enzymes. Many enzymes, usually most of the enzyme have a suffix as. For example, if some enzyme works on urea, then we can call it urease. If some enzyme work on amylose, we can call it amylase. But these are not fixed. There are a lot of exception here. Like some enzymes, for example, glucokinase acts on the substrate glucose, that's why we called it glucokinase. Or sometimes they are transferring molecule like aminotransferases, because they are transferring amino group. So these are not fixed actually. IUBMB, International Union of Biochemistry and Molecular Biology, they introduced nomenclature system which is called EC. EC stands for Enzyme Commission number. Here EC number must consist of 4 digit. Each EC number must contain 4 digit, and is first digit class, second digit subclass, third digit will be subgroup, and fourth or final digit will be the number of particular enzyme in the list. For example, catalase EC number 1, 11, 1, 6. That means 1 stands for the class. I will discuss later about the enzyme class in second part. That enzyme has 7 classes so far, and the first class is catalase. So that's why first class it will be the class, and second one first class is oxidoreductase, and 11 number is the subclass. Why these are in this group? Because these catalase act on peroxidase as acceptor. And then subgroup is 1, the third digit is subgroup. Why 1? Because it acts on peroxidases. And 6, that is the number of particular enzyme in list, catalase, this is in 6 number. And same in case of alcohol dehydrogenase, this one is 1 for class 1 oxidoreductase, and subclass acting on CHOH group, and then subgroup because it's act with NAD or NADP as acceptor, and its class name alcohol dehydrogenase. And we can find it in Brenda details also in the website of International Union of Biochemistry and Molecular Biology website. Here I give the website. If you are interested, you can have a look here. So let's come into the summary. First enzyme, they are biocatalyst. They accelerated the reaction rate, and they required a very small amount, but they remain unchanged after the reaction. These 3 must be included in an enzyme. To be an enzyme, it must accelerate the reaction, and it required in small amount, and it remain unchanged after the reaction. And most of the enzymes are protein, but there is some exception which we call ribozyme, which is the catalytic RNA molecules. Then enzymes lower the activation energy. As we know, enzyme needs substrate needs activation energy to transitional state, and then they release the free energy to reach the product. So here the enzyme lowered the activation energy or barrier so that they speed up the chemical reaction. And catalytic mechanisms, mostly acid base catalysis, covalent catalysis, metal ion catalysis, and also stabilizing the transitional state by using enzyme. And change of the reaction rate of enzyme, that is the Q10. Q10 is the change of that is the coefficient 10 Q10 for enzyme, change of relative reaction rate of an enzyme at two different temperature which are differing and 10 degrees Celsius. That is, the temperature difference must be 10 degrees Celsius, and the 10 degrees Celsius enzymes reaction rate known as the Q10. And what is apoenzyme? The protein part of an enzyme known as apoenzyme, and non-protein part of an enzyme known as cofactor. And what is holoenzyme? Holoenzyme is the total protein part and non-protein part of an enzyme known as holoenzyme. What is prosthetic group? Prosthetic group is tightly attached. Bear in mind, it must be tightly attached, permanently or covalently, with organic or inorganic. And in case of coenzyme, this one is loosely attached and must be organic. So coenzyme is loosely attached and organic, and others may be organic or inorganic. And mechanisms of enzyme action. Enzymes action mechanism we can classify in two ways: lock and key model, where the active site is fixed, they are rigid, and induced fit model. Here active site is loose, active site is flexible, and they allow the substrate to fix and also accommodate and also change the conformation of the active site in case of enzyme. And Enzyme Commission number has 4 digits. First one is class, subclass, subgroup, and the number of the enzyme in list. Thank you very much for watching.